Hello everyone, let's learn about normal sexual development. So, to learn about the normal sexual development, we need to learn two important things first. First of all, we will be talking about the PGCs or the primary germ cells, and then we will be talking about the genital ridge. Because these are the two structures, these are the two important structures from which the gonads are going to develop. The primordial germ cells, they are derived from the primordial germ cells are derived from the epiblasts. They are derived from the epiblasts and they are present in the yolk sac. Rather, we can say that they are present in the wall of the yolk sac. What happens that during the 5th week of the intrauterine life, the primordial germ cells will migrate through the dorsal mesentery to the region of genital ridge by amoeboid movements. So let us first understand the primordial germ cells first. As we learned that the primordial germ cells, they are derived from the epiblast. Epiblast, if we try to recall that whenever we learned about the formation of the bilaminar germ disc, where the cells here they were known as the epiblast and the cells below they were known as the hypoblast. So these are the epiblasts, which will later many things later. Let's not talk about this. Then these primary germ cells, they are present in the yolk sac. I will draw a diagram so that things get clear. So this is the embryo and here we have the yolk sac. The yolk sac, it means the embryo will be divided into the foregut, midgut and hindgut. Now what is this genital ridge? Okay, before that we have learned that this primordial germ cells, because this is the yolk sac, so the primordial germ cells, they are present inside the yolk sac. It is present in the walls of the yolk sac. Now it is going to what did we learn is that it is present in the yolk sac, and in the 5th week of the intrauterine life it is present in the yolk sac, and in the 5th week of the intrauterine life the PGs, the primordial germ cells, will migrate through the dorsal mesentery to the region of the genital ridge by the amoeboid movements. So we are left with the understanding of the dorsal mesentery. We learned that the PGs, they are present in the walls of the yolk sac. Now what is this dorsal mesentery? The tubes which are present, these are the tubes, means the intestines and all. So these are the tubes here, and the tubes are connected to the posterior wall with the help of the fold of the peritoneum. And what is that? That is the dorsal mesentery. Let us assume that this is the dorsal mesentery, and the primordial germ cells so this is the dorsal mesentery, and the primordial germ cells from the yolk sac via the dorsal mesentery, it is going to reach this region. Now this region, as I told you, that this is the genital ridge. Now let us explain what this genital ridge is. So did you understand the migration, or have you all understood the migration of the primordial germ cells, which was initially present in the yolk sac? In the 5th week of the intrauterine life, it is migrated through the dorsal mesentery to reach the genital ridge. Okay, now let us understand what this genital ridge is. If we again look into this diagram, this genital ridge, let us first of all understand what is the definition of the genital ridge. In the 5th week of the intrauterine life, again here also it says that in the 5th week of the intrauterine life, the coelomic epithelium in the medial surface of the mesonephros proliferate to form the genital ridge. What is this? To form the genital ridge. So let's learn what the new words that we learned here is that we in the 5th week of the intrauterine life. So we have learned that in the 5th week of the intrauterine life as well, the coelomic epithelium which is present in the medial surface of the mesonephron will proliferate to form the genital ridge. Now what is this mesonephrons? First of all, let me just magnify this posterior abdominal wall or the dorsal wall of the embryo. If this is the dorsal wall, there are two important structures which are present in the dorsal wall. One is our very well-known dorsal aorta. So this is the dorsal aorta. Another important structure which we discuss is the mesonephrons. So this is the mesonephros, and this is the dorsal aorta. Now the epithelium which is lining in the medial side, this is the mesonephron. So here there are the cells which are lining in the medial side of the mesonephron. So the cells which are lining in the medial side of the mesonephron, that is known as the coelomic epithelium. And it is this coelomic epithelium which is going to proliferate. When this proliferate, it is going to form, suppose it is proliferating like this. So it is going to form a bulge on the posterior abdominal wall, which is known as the genital ridge and nothing else. So it is forming a bulge. This is the medial part of the mesonephros which is forming a bulge on the posterior abdominal wall, which is known as the genital ridge. So this is only known as the genital ridge. So let us just understand this thing once again, that I told you that for understanding the normal sexual development, we need to understand two important structures. First of all, there is the primordial germ cells. There is the primordial germ cells and the genital ridge. Now the primordial germ cells, they are derived from the epiblast. They are present in the yolk sac, and during the 5th week of the intrauterine life, these primordial germ cells will migrate from the yolk sac to the posterior abdominal wall with the help of the dorsal mesentery. And to the where in the posterior abdominal wall? To the genital ridge region in the posterior abdominal wall. And how is it going to go? It is going to go with the help of the amoeboid movements. Then we learnt about the genital ridge. What is the genital ridge? Again, in the 5th week of the intrauterine life, the coelomic epithelium which is covering the this is the coelomic epithelium which is covering the medial side of the mesonephros. This is the mesonephros. So the coelomic epithelium which is covering the medial side of the mesonephros is proliferating to form the genital ridge. Okay, learning these two chapters is very important, because we are now going to understand what happens and why are these important. So okay, so where is it? So that means the genital ridge, it is being formed from the mesonephrons. Important thing to note is that this genital ridge, as I told you, that inside the genital ridge only. So that means by 5th week of the intrauterine life, inside the genital ridge, now in the genital ridge, or you can write now, the genital ridge has, what does it have? It has the primordial germ cells which have migrated into it, and it also has the coelomic epithelium. The coelomic epithelium, it is basically formed from the mesonephron, and this mesonephron is a part of the intraembryonic mesoderm. It is a part of the intermediate mesoderm, or rather you can say that it has developed from the intermediate mesoderm. Just a quick recapitulation. We have learnt that suppose this is the notochord, this is the paraxial mesoderm, this is the intermediate mesoderm, and this is the lateral plate mesoderm. Paraxial mesoderm, intermediate mesoderm, and this is the lateral plate mesoderm. So the intermediate mesoderm, which is present in the lumbar region, like it is the intermediate mesoderm. Let's not talk about that, but the intermediate mesoderm is also present in the form of this tube, and what will happen, it is going to thicken to form the nephrogenic cord. This intermediate mesoderm will thicken to form the nephrogenic cord, and it is the different segments of the nephrogenic cord which we talk about, it will get separate. These are the different segments. One is the mesonephros, then we have the metanephros. This is going to form the kidneys. Okay, so this is just a small orientation of why are we telling that the mesonephron is originating from the intermediate mesoderm. Alright, so genital ridge is having the PGCs and the coelomic epithelium. This is a question. Okay, next, now why are we reading the genital ridge in so much detail? What is the genital ridge? Yes, so the genital ridge was made up of the coelomic epithelium, and it had the primordial germ cells. Now remember that it is from this coelomic epithelium only that numerous finger-like processes will be produced. Or if we just don't think too much and we just need to understand what is it, then remember that it is from the genital ridge only the primitive sex cords will be formed. That is, the primitive sex cords will be formed, and these primitive sex cords, they are bipotential. They are bipotential, which means that they have the capacity to get converted or to differentiate either into the male gonads or into the female gonads. Now, whether it is going to differentiate, so from the genital ridge, therefore it is going to differentiate to form the primitive sex cords. It is because of the formation of the finger-like processes from the coelomic epithelium. Now, these primitive sex cords which are formed, they are bipotential and they have the capability to get converted to differentiate into either the male gonad or the female gonad. So, who is going to determine that? It is this. That we know that the males have the chromosomal constitution is XY and female is XX. The Y chromosome in the short arm of chromosome number Y, SRY gene, which is also known as the TDF, which is the testis determining factor. SRY gene, which stands for sex-related gene, which stands for sex-related gene on chromosome Y, and TDF stands for testis determining factor. Testis determining factor. Okay. So this SRY gene or the testis determining factor, this particular gene, where is it present? It is present in the short arm of chromosome number Y. The short arm of chromosome Y. Now, what is the speciality of this gene? That if this SRY gene is present, what it does is that it is going to activate SOX9, and this will result in the differentiation of this primary sex cords into testis. Right? It is because of the presence of this SOX9 gene means the SOX9, which is produced by the SRY gene, and this SRY gene is present in the short arm of chromosome Y. So males, they're having the chromosomal constitution is 4661. Y is this Y chromosome. In the short arm of the chromosome Y, what is present? This SRY gene is present, which is going to stimulate SOX9, which will result in the differentiation of the primitive sex cord into testes. Now testis has two type of cells, that is the Sertoli cells and the Leydig cells. So as we know that it contains the Sertoli cells and the Leydig cells. So the Sertoli cells, it secretes under the influence of the FSH. The Sertoli cells are going to secrete a hormone which is known as the anti-Mullerian hormone. Now this anti-Mullerian hormone, what it does is that, so we are talking about the male part first. This is known as the anti-Mullerian hormone or the Mullerian inhibiting substance. Anti-Mullerian hormone, anti-Mullerian hormone or the Mullerian inhibiting substance. Inhibiting substance. What it does is that this is going to cause the regression of the Mullerian duct, the duct which is responsible for Mullerian or the paramesonephric duct, and this is because this paramesonephric duct is responsible for producing the female reproductive tract. It regresses the Mullerian duct and it leaves the remnants of which are important to remember, that is, it forms prostatic utricle, hydatid of Morgagni, and one more thing, that is appendix of testis. So not only does it cause the regression of the Mullerian duct, but it also helps in the abdominal descent of the testis. Okay, so this is what the Sertoli cells do. The Leydig cells, they are going to produce testosterone, and this testosterone is produced in the 8th week of the intrauterine life. The function of the testosterone, it is as follows. First of all, the testosterone, because we saw that the Sertoli cell, it produced the anti-Mullerian hormone, which suppressed the growth of which duct? The Mullerian duct. It suppressed the growth of the Mullerian duct or the paramesonephric duct, which was supposed to develop into the female internal genital organs. Now, because the paramesonephric duct has been suppressed, so this testosterone is going to stimulate the growth of the mesonephric duct or the Wolffian duct. Now, because this duct is going to grow, it is going to grow into what? It is going to grow into the male internal genital organs. So this is how the male internal genital organs are going to develop. That is the seminiferous tubule, epididymis, all these structures. Next, what the testosterone does is that under the influence of the 5-alpha reductase enzyme, the testosterone is converted into dihydrotestosterone, and it is this dihydrotestosterone which is responsible for causing the masculinization of the external genitalia. The external genitalia during the stage of development, this external genitalia also has the bipotential capability to get converted into the male or the female external genitalia, but under the influence of the testosterone, with the dihydrotestosterone is formed, which causes the masculinization of the external genitalia. Okay, the other functions are that it is going to have a negative feedback on the GnRH hormone, due to which the GnRH will decrease, and the FH and LH are also going to the FSH and LH are also going to decrease. And the other function which the testosterone has is that it causes the descent of the testis, the scrotal descent. Sertoli cells did something. It did the abdominal descent of the testis. Testosterone causes the scrotal descent of the testis. Here we learned this is the function of the testosterone, and it is with the help of this testosterone only that we saw that the male internal genital organs are being formed. The external genitalia, they are getting converted into the male one, that is, the masculinization is happening, and it also causes the scrotal descent of the testis. So with this we complete the development of the male genital organs of the male reproductive system. Now if we talk about the female reproductive system, then we know that it is very clear. Female, now we talk about female, which is 46XX. Now because there is no Y chromosome, that is why there will be no Sertoli cells. First of all, let's not go to Sertoli cell, because there is no Y chromosome. Obviously there will not be the SRY gene. If there is no SRY gene, there will be no Sertoli cell. If there is no Sertoli cell, there will not be any the anti-Mullerian hormone will not develop, means it will not be produced. So that means what will happen, the Mullerian duct grows, and it is going to form the female internal genital organs. Along with that, because no Sertoli cells are present, anti-Mullerian hormones are not produced. Similarly, no Leydig cells are present. And because no Leydig cells are present, so no testosterone. And if there is no testosterone, so there will not be masculinization of the external genitalia. So no masculinization of external genitalia. So what will happen? Female external genitalia will be produced. External genitalia is produced. So this is how that so in the absence of the Y chromosome in females, how the female internal genital organs are formed, and how the female external genitalia is produced because of the absence of the SRY gene. Now one more thing is that now because there is no testosterone was causing the growth of the Wolffian duct. So if there is no testosterone, this is the testosterone, I'm sorry for my handwriting. So because there is no testosterone, so one more thing is going to happen, that is the Wolffian duct is not going to form. So Wolffian duct will regress. Wolffian duct regresses to leave the remnant, and the remnants which are produced are epo-oophoron, para-oophoron, and the Gartner's duct. So this is how, with this we complete the development of the male and the female external genitalia. Thank you for your patient listening.